introduced, I should start, right? So, what I want to share with you today is some of the evidence for exercise being good for you and how it works. What does exercise specifically do? This slide I'm supposed to show, that's me. And my disclosures are basically I've had grant funding and I serve on an advisory board of a center, but I don't have any conflicts. So this slide is one that I stole from some advertising a couple of years ago that shows kind of the evolution of humans. And where are we going? And it looks like that we're taking a step back from a health point of view. We are having lots of food and very sedentary, very inactive. So we should do something about that. And this is the science of sweat. And this is actually the cover of Forecast magazine, which is a lay publication by the American Diabetes Association from a couple of years back. And basically they say, you know, exercise, it's free. Most of us don't do enough of it. We all know we should. From a diabetes point of view, exercise does a lot of good stuff. It's like the ideal drug. Improves your blood sugar, it cuts down on some of the long-term risks of diabetes, cardiovascular disease in particular. So I'm going to go way back in history and we're going to talk about these three guys, Herodotus, Hippocrates, and Galen. Herodotus at 480 BC is considered the father of sports medicine. And he was a physician who actually taught Hippocrates. And he was the first to actually write about exercise as a treatment for disease, for treating disease and preventing disease. And he advocated everybody should exercise regularly and you should eat a very healthy diet, avoid fast food. He actually was involved in the training of elite athletes as well and had strength training and various aspects of exercise training to improve performance. And that's kind of the sports medicine aspect of it. Hippocrates is somebody we're all familiar with. Galen is also somebody we should be familiar with, but Galen came several hundred years after these two guys. And Galen was also a Greek physician, but he was a Greek physician who went to Rome and practiced medicine there, and was physician to several emperors of Rome. He was widely accredited with kind of a great deal of knowledge. And one of the things that he did that would make us who write our CVs very jealous is that he actually wrote over 300 essays and treatises on a whole bunch of stuff related to medicine. His writings have been used and taught for 1,000 years after he wrote them. But he's commented, this is a quote from one of his treatises, he's commented on what exercise is. If you actually are breathing fast, that's probably exercise. Some of us can do more without breathing fast, but 3 things result from exercising. Hardness of the organs, we're talking about muscle hypertrophy specifically. And then intrinsic warmth and accelerated movement of respiration. He's talking about the actual exercise itself. He did a lot of things related to circulation and nervous system. And these things were quoted, as I said, over 1,000 years following when he wrote them. He never dissected humans, but he dissected multiple animals, including pigs, to come up with the circulatory system and the nervous system. That's the really old history, but What I want to share with you now is some of the evidence that exercise is good for you. And we're going to talk about mortality. And you're all familiar with the Framingham Heart Study, and this is data from the middle 90s where they looked at reported exercise in men. These are the older men in the study. Looking at physical activity quartiles, the men who exercised the most compared to those who exercised the least is about a twofold difference in overall cardiovascular mortality. So in the Framingham Heart Study, it's very clear that exercise was beneficial. And this study is one that I like a lot. It's really pretty interesting. And this comes from Olympians from Finland. And this is a study of over 40 years following up on people who were Olympians from Finland. And the controls in this study were people who were contemporaries of the athletes. And the contemporaries, in order to be a control, they were people who were age-matched who entered into the military in Finland. So they were considered to be healthy young people. But as you can see, there's a really dramatic difference. Maybe I should do it with this. Does green work? Not very well. I guess I'll go back to the mouse. There's a huge difference here in overall mortality, cardiovascular mortality, and cancer mortality among runners who were Olympians compared to their contemporary controls. If you look at group sports like soccer or hockey, there's also benefit in cardiovascular and cancer. If you go all the way to weightlifters, and these people tend to be bigger, bulkier men, there wasn't any significant improvement in cardiovascular mortality, but still there was cancer mortality that was significantly lower. And you can see by the numbers at the bottom here that these are substantial numbers, statistically very impressive. So distance runners have the best outcome. This question.
Is it significant, the difference in cardiovascular mortality between weightlifters and control?
I heard significance, but I didn't hear the whole question.
Is there a significant difference between cardiovascular mortality between weightlifters and control or not? Is that because I saw it was high in weightlifters compared to control? And is that significant or not?
I could not hear everything you said. Let's see if I can wing it backwards.
So he's saying that the weightlifters, the cardiovascular mortality is higher than the control.
Oh, yeah.
Is that difference significant or not?
Yeah, I do not know the answer to that. They didn't have standard errors. Within the paper itself, we're looking at long-term follow-up of athletes until they're in middle or older age, and probably not just the sport that they engaged in 30 or 40 years ago, but what they did in between Olympics and the current time is important. And what they found was, and it's described in the paper, is that the distance runners were much more likely to continue exercise lifelong, excuse me, compared to weightlifters who did not. So the actual event sort of designates what people did for the previous 40 years before the mortality data. I'm not sure. Oh, I guess I did answer the question. I don't know about statistical significance, whether it's more or not. So that was overall mortality, cardiovascular mortality, and cancer mortality.
So just going back to the previous chart, did anyone possibly
Are there other differences to the question besides the actual exercise?
What's the difference in cardiovascular mortality between the weightlifters and the distance runners?
Yeah, and that was not dealt with effectively within the paper, but the question is there something else besides the exercise? Do they have a genetic predisposition based on some other factors? And I would say that weightlifters are in fact different from distance runners, physiologically and biochemically. So it could be that having a higher percentage of strength fibers in your muscle, your skeletal muscle, puts you at some higher risk for other risk factors like diabetes or hypertension. But that's, again, that's not clear. You have a question?
Yes, sir. Well, weightlifters, they develop LVH because you actually induce hypertension whenever you're lifting weights like that. Like extreme weights, your blood pressure goes up, and so at least that's what, when I've seen that, they actually would have LVH more than a normal person would. So
Well, if you're looking, I think that it's an interesting thing we can talk about for probably an hour about what the hearts look like on athletes. And weightlifters and distance runners have LVH.
I mean, there is an entity in this called athlete cardiomyopathy, but I'm not sure if it's more in weightlifters than as distance runners. I mean, definitely weightlifting causes vasoconstriction, increase the afterload. You do cardio more cause vasodilation, decrease the afterload. But these are related things. I don't know if any studies that.
I think the answer is a little bit easier, honestly. Weightlifters just take a lot more calories in to maintain that muscle mass. A lot of calories. No.
All right. We're going outside of what we're supposed to be talking about today, but your point is that calories in are substantially more in athletes than they are for the rest of us. And weightlifters tend to have a body composition which is not as favorable as a soccer player or a distance runner, where they'll have lower body fat. And that may be a big part of it and puts them at risk for other things. Yes?
I can understand the decrease in cardiovascular mortality by exercise. But how does the exercise will affect the cancer mortality?
How does it affect what? Cancer?
Cancer mortality?
That's a good.
I can understand the cardiovascular mortality.
Again, it's people who are probably smokers, right?
Yeah. All right. What I think you guys are actually answering the question yourselves, and that is, I've seen a study where mortality, cardiovascular mortality is higher in people who don't wear seatbelts. What? And it's choices you make in health-related behaviors. So, do weightlifters smoke more? I would say that runners smoke less. So, I mean, it's more complicated than just exercise. Our lives are more than the 30 minutes we do on the treadmill every day. How many do that? We all should. So cancer mortality is fascinating here, and mechanisms are unclear. But part of the paper did, in fact, as I already alluded to, the people who were runners exercised throughout their lives more than the people who were weightlifters. So let's move on to prevention and treatment of diabetes. And how does exercise work with diabetes? There's only one data slide that I'm going to share with you, and this is kind of the other side of the Women's Health Initiative. This is male health professionals, and these are data that come from 20 years of questionnaires sent out every 2 years, and there's over 500,000 person years reflected here. But as one might be expecting, if you exercise a lot, 150 minutes per week, which is one of the recommendations that we'll talk about later, you cut your risk of diabetes in half. But of interest is also that if you do it less, there is some benefit. If you do it for 60 minutes, you cut it by a third. So regular exercise, at least this is questionnaires filled out, not actually observed exercise, so it's probably an overestimate of what people actually do. But regular exercise helps to prevent diabetes and everything that goes with it. So we're going to move on to another category where exercise is used as a treatment, and that's frailty of the elderly. And the frailty of the elderly is an interesting topic unto itself. What I'm going to show you in the next, oh wait a minute, I got to remember what order the topics are. What I'm going to do before I share with you the data from the studies that we've done here is we're going to talk about physiology and biochemistry of exercise. And this is going to be a brief refresher course to remind you of the things you probably heard when you were in medical school. This is a transverse section of skeletal muscle from humans that's been stained for mitochondria. The green represents capillaries that surround muscle fibers. And there are basically three kinds of fiber type that we work with today. There are older literature that suggests there are many more types, but three is what we're talking about in most studies that we work on these days. Type 1 fibers are slow twitch, endurance, oxidative fibers. Type 2 fibers are fast twitch, and 2X are strength fibers, pure strength fibers. And they're categorized as glycolytic rather than oxidative, mitochondrial poor rather than mitochondrial rich. And most of us have about 50-50, 50% type 1s and 50% type 2s that are subdivided between 2X and 2A. The 2A fibers are kind of a mixture of categories of characteristics of type 1 and type 2X. So they have some more oxidative functions, some more mitochondria compared to 2X. These are slides from studies that we've done where we, as part of the studies, we have to characterize what the fiber composition is prior to training and then post-training. And type 1 fibers are stained, these are all antibody staining techniques. Type 1 fibers in the bright field stain very darkly blue. Type 2X fibers stain pink or red. And the type 2A are kind of a mixture, so they're purple. In immunohistochemistry, where we have three specific antibodies, type 1 in this category, this technique is green, 2X is bright red, and all the rest that are blue are 2A. So these things are important as we look at response to exercise training, which I'm going to share with you after we finish this basic physiology biochemistry background. This represents a muscle. This represents a fascicle, which is made up of actual muscle fibers. And each muscle fiber has multiple contractile apparatuses called sarcomeres. And sarcomeres are the units that contract. The sarcomere is made up of thick filaments and thin filaments. The thick filaments are myosin, heavy, and light chains all twisted together. The thin filaments are actin, troponin, and tropomyosin. When the sarcomere shortens, the thick filaments don't move, but the thin filaments do move, and they are pulled along by the thick filaments in order to shorten. And this is a more detailed schematic showing the thin filaments containing the troponin and tropomyosin. Troponin interacts with calcium to initiate the process. And what I'm going to share with you is a 4-minute YouTube video. Which I think is pretty cute. And this is assuming we're going to have
You use
sound.
muscles every day to do
I
activities.
was promised sound.
This woman is using muscles to breathe, circulate blood, and move her hand to take
I
notes.
don't hear anything.
Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of fibers.
I'm going to stop
Muscle
it for a
fibers are
second and
long
see if
cylindrical
Daniel can
cells
help.
containing several. Nuclei. You use muscles You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
So, I didn't want to go to the next one.
Are you sick of flipping paper? Are you sick of flipping paper?
So, let's see. Are we still in? Yes, we are still in. So that was 4 minutes that tells everything you need to know about muscle contraction physiology. But what I'm going to share with you now is data from studies that we've done over the last several years using exercise as a modality to prevent diabetes. And within skeletal muscle, there are really 3 different pathways that we're very interested in. There's the insulin action pathway, which starts with an insulin receptor and goes through multiple steps, but actually then ends with translocation of glucose transporters to the cell surface. There's a hypertrophy pathway, and the hypertrophy pathway is a protein synthetic pathway that helps provide increase in muscle bulk. And then mitochondrial biogenesis. And these 3 pathways are not entirely independent. They interact. And one would like that the therapeutic approach that we use that interacts with any or all of these pathways could help the one in the middle, which we are most interested in making insulin work better, to make insulin resistance better, to make your risk for diabetes better. The hypertrophy pathway is stimulated primarily by strength training, and the mitochondrial biogenesis is primarily endurance training. And diet, that is, weight-reducing diet, impacts the insulin resistance that we're most interested in metabolic syndrome patients. And these are 3 different studies that we've done over the last several years. One is resistance training for 8 weeks, where we had people do weightlifting primarily. We did a separate group of people with endurance training where they trained on stationary bicycles. And then the third one was to do resistance training, but to go beyond 8 weeks and actually double the time and the intensity of the training. The protocols were all metabolic syndrome patients who had not yet gotten diabetic. Each of the protocols had control subjects who were lean, also sedentary, but lean, otherwise healthy subjects. We had men and women in every protocol, and the protocols were all progressive, so they start low and they increase the intensity throughout the 8 to 16 weeks. We did not allow weight loss because we wanted what we measured to reflect exercise training, not a combination of exercise training and weight loss. So we didn't allow weight loss. And finally, we did some quantitation of insulin responsiveness, which is an insulin infusion and looking at see how well insulin works on glucose uptake in the whole body. And this slide summarizes the characteristics of the subjects. This is about 50 people altogether. 30 of them had metabolic syndrome. And of interest, as you can see here, is the body mass in the metabolic syndrome people is considerably higher. We're looking at over 100 kilos. Body fat content, actually almost 30% in our controls, but that's a mixture of men and women. BMI of the controls are less than 25. And our metabolic syndrome people averaged about 35, so moderate obesity. VO2 max is a measure of aerobic fitness. The higher the number, the better. So our controls are running at about 30. Our metabolic syndrome people are running considerably less than that. And of interest, and this is not exactly what we expected though, is that looking at overall strength prior to training, is our controls and our metabolic syndrome people were essentially the same in overall strength. And the next several slides are just some pictures of subjects that were participating in. This guy is one of the graduate students in the earliest study. And this is one of the subjects who's warming up before actually going into the resistance training. And this is one of our subjects. And the resistance training is lifting weights mostly. There are other things that we do as well. And these are a couple of subjects who participated in the endurance training. The one on the left is a control subject, and this is a metabolic syndrome. They happen to be married to each other, so that it was good to get two of them together, a control and a metabolic syndrome. This is another subject doing one of the testings that we had to do on every subject. This is looking at the strength, and she is pulling against a stationary bar and exerting maximum force. She's standing on a force plate that actually quantifies how much force she's generating. And this is, again, our graduate student who is trying to get them to work as hard as can possibly be done. This is another subject, and one of the things that we measured on all of our subjects before and after was jump height. And jump height is one thing that our exercise science people really like because it correlates with performance of athletes, is jump height. And the VO2 max, measuring basal metabolic rate and maximum oxygen uptake, is done here with the equipment. Again, 2 exercise science graduate students participating in the studies. And this slide kind of shows just the overall perspective on the intensity of the training throughout the 8 weeks. We start off kind of light and then gradually build the workload throughout the 8 weeks. And then the final week of the 8 weeks is an easy week, which we then move on to the measurement of strength and VO2 max and muscle biopsies again. Everybody got a muscle biopsy of vastus lateralis before and after the study. The 16-week people actually got 3 muscle biopsies, one at baseline, one at 8 weeks, and one at 16 weeks. The muscle is maybe 50 to 100 milligrams of muscle tissue, which is quickly processed by our team who is sitting over there right now, where they take a portion of it and process it for histology, which has to be processed separately from the rest of it, which is then, the rest of it is frozen in liquid nitrogen for biochemical quantifications. So what were the results? This is body composition, and it's comparing endurance training and resistance training, strength training. And remember, we said no weight loss. And what we found was in the strength training, they actually changed their body composition in the way we'd like them to do it. They increased their muscle mass and did not change their fat mass. And because it's a paired study, everybody increased a little bit, and it was highly significant. So both controls and the metabolic syndrome people increased their muscle mass during strength training. The endurance trained subjects didn't change. And you can see graphically the body composition between muscle and fat, or lean body mass and fat mass, is quite different in the metabolic syndrome compared to controls. And one would say, and many people would criticize our controls. Our controls actually have a lot of body fat, even though their actual weight for height was fine. And it has to do with sedentary behavior over at least a year before the studies were done. This graph shows the strength changes that occurred with subjects. You can see on the left, These are the people who underwent strength training and they got a lot stronger. Both controls and metabolic syndrome people, it really worked. It worked very well. Endurance training is bicycle training, and it's really intended to improve endurance. And in fact, it did not significantly change strength, kind of as we would have expected. Now, aerobic fitness is the VO2 max, and we would have thought that endurance training would increase the VO2 max, and it did on the right. And we didn't expect it to do that on the left in the strength training, but in fact it did there too. So everybody improved their VO2 max, their aerobic fitness. And finally, well, maybe not finally, but insulin responsiveness, which was our primary outcome variable, we saw a little bit of improvement in the sedentary controls with strength training, but really no change with metabolic syndrome and really no change in endurance trained people. So no weight loss allowed, and exercise training did not have an impact on insulin responsiveness. As you can see, the insulin responsiveness in the metabolic syndrome people is less than half what the controls have. And actually in the second study, it was more like 35% of the control subjects. So the biochemistry is the next several slides, and we measured insulin receptors in skeletal muscle in those muscle biopsies. And in the resistance-trained people, there was a substantial increase in insulin receptors. So they got stronger and they got a lot more insulin receptors within skeletal muscle. And if we look across the board for endurance trained and strength trained, everybody got a lot more insulin receptors, substantially increased the insulin receptors. So insulin ought to do more, right? The other end of the insulin action pathway is glucose transporters. And what we found was, in fact, similar to the insulin receptors is, I guess we'll go back a little bit here. The GLUT4, which is the insulin responsive glucose transporter, substantially increased. And in fact, across the board in all the studies we did, GLUT4 changed the most of anything we measured. GLUT4 substantially increases with exercise training of any kind. We measured mitochondria in several different ways, including ATP synthase, which is really the end of the chain that generates ATP within mitochondria. And then mTOR, which is the principal point of control for muscle hypertrophy and protein synthesis within skeletal muscle. And what we see here is strength training did exactly what we expected it to. Remember, the body composition showed muscle hypertrophy, strength improved, protein synthesis actually increased, and the mTOR was activated substantially by strength training. With endurance training, where we didn't see change in body composition, mTOR was not substantially activated. So just to summarize all that, the insulin clamps, the insulin infusion stuff didn't change with either resistance or endurance training in spite of insulin receptors going up and glucose transporters going up. And there were other things that I showed you, the mitochondria and the activated mTOR. But where's the problem? Insulin receptors went up, glucose transporters went up. Why didn't we see more? Well, there are other parts to the insulin pathway. And what we anticipated was insulin receptor substrate 1, which is the first step beyond activation of tyrosine kinase within the insulin receptor, is where the problem lies. And I'm not going to show you all the data, but we did in fact demonstrate that that is the problem in metabolic syndrome people, is that the IRS1 is hyperphosphorylated in inhibitory sites, so that the insulin activated signal is not stopped, but it is much diminished in metabolic syndrome subjects compared to controls. So IRS1 seems to be at least the principal site of insulin resistance, and it is not affected by either endurance or resistance training. Remember, I said diet, and we prevented weight loss. So we're going to start talking now about stuff that we didn't do, but other people have done, and that's exercise training and frailty in the elderly, which is one of the things that we are recommending exercise training for. Now, why do we have frailty in the elderly? As we get older, there's a loss of muscle fibers. There's a decrease in muscle fiber size, and the loss of fibers is primarily strength fibers, type 2 fibers. And this is a publication back in the 80s, actually, from the Karolinska in Stockholm, where they collected muscle from people who died accidental deaths of various ages. And what they found was that as age increases, and you can see the older ages we're talking about in the 80s, as age increases, the number of muscle fibers, and this is the number of fibers in the vastus lateralis, the whole cross-section of the muscle, declines to to less than half of what it had been in young men. And each of the fibers is smaller. And all this is called sarcopenia. So what can you do about it? Maybe we should start with why does it happen? And this is a graph that is data from NASA. And this was done when I was actually working with the people at NASA when I was in Texas, where they actually went around through NASA in Clear Lake, and they asked every employee to have VO2 max done. And almost everybody did it. And what this actually shows is the VO2 max, and the dots are women. There are over 400 women who agreed to doing a VO2 max, is there's an age-related decline in VO2 max. And VO2 max correlates with physical activity. So this is a surrogate for exercise history. And it basically says that both men and women decrease their physical activity over time. And that probably is the single most important reason we have sarcopenia in the elderly. There are other reasons, though. I mean, one of the important ones is testosterone or estrogen steroid hormone changes that occur as we get older. So what can be done about it? There have been quite a number of studies that have been done over the last 20 years where they've tried to have interventions which improve the strength or improve the risk of falls in older people. And this is a study that was done with 99 nursing home dwelling subjects who were in the nursing home actually because of mobility problems. And they randomly assigned them to nothing or to strength training in the lower extremities. And there was also a protein supplement that was given. But as you can see, there is a dramatic benefit in this variable called stair climbing power, where they actually did force plates similar to the project that we did, and showed that resistance training in the lower extremities substantially improved older persons' lower extremity strength. So what I'm going to do now in the next couple of slides is talk about innovations in exercise. And the innovations are here: HIT and SIT and NIT. And I suspect that many of you have heard of HIT. And that's high intensity interval training. And we're calling it innovation, but it's really kind of not that new. There have been lots of publications from the last decade on HIT. It started off with a single group at McMaster in Hamilton, Ontario, and then moved on to several other groups, kind of confirming the benefits of high-intensity interval training. SIT is sprint interval training, which is really just a variation on HIT, where people are sprint and relax and sprint and relax. And finally, the MICT is kind of what we've been doing forever, and that's moderate intensity continuous training. So that's not an innovation at all, but that's to which all of these things are compared. And that's what this is moderate intensity. Brisk walking, that's moderate intensity continuous training. But this slide is an example of a HIT program, which is high intensity interval training. And this is on a stationary bike, but there are others you can find online that talk about treadmill and other modalities for exercise. But it starts off with a few seconds of higher intensity exercise, followed by relaxing, doing less. And the high intensity is running at 75 to 90 RPMs, and the recovery is at a lower rate of 55 to 65. And then it just keeps going up. 15 seconds alternating, 30 seconds alternating, 60 seconds alternating. But 60 seconds is as long as the high intensity goes, followed by a similar period of relaxation and recovery. And this whole project, the whole protocol, only takes about 10 minutes. So what high intensity interval training has done is that it's said that this resistance training like we did for 8 weeks, where you gradually improve or increase the intensity, is inefficient. It's an inefficient use of time. And the high intensity training with much less time invested achieves similar results. So this is kind of where a lot of training protocols are going for athletes and for health in general. So all that shared with you, what are we supposed to tell when our family asked us what kind of exercise should we be doing and how much? And this is a paper that came out, I guess it's now 5 or 6 years ago, but it's from the American College of Sports Medicine, and every few years they come out with a recommendation as to what kinds of exercise and how much we should be doing. And that's what this is, taken from that paper. We're talking about the 30 minutes, 5 days a week, which we've been hearing in the media for a long time, but they've added things to it. 30 minutes, 5 days a week of moderate intensity cardio, that's the MICT. But you should be adding vigorous intensity cardio to that, and do that maybe 3 days a week for a shorter period of time. And you have to have resistance training. You can't just do cardio. You've got to do resistance training 2 to 3 times per week and use as many muscle groups as you can do. And there are 10 or 12 major muscle groups, so you're going to be doing sets and reps and involve many different muscle groups. And they're recommending that there should be some flexibility, some stretching that's part of this as well. So what's it all for? Well, I've shared with you some of the data that supports this, that regular exercise, including both aerobic and strength training, decreases cardiovascular mortality, decreases cancer incidence and mortality, decreases obesity, decreases diabetes risk, decreases falls and mobility impairment in the elderly. And then finally, which I didn't show you any of these data, there's some evidence that regular exercise can delay or prevent cognitive decline. And that's the end. And we've filled up the hour and we have questions.
How did they correlate activity in cancer incidence?
Say again, please.
So how would they correlate cancer incidence with the increased exercise?
What's the mechanism of the decrease in cancer incidence and mortality? And we kind of answered that earlier with questions that came from the audience. And it's unclear. There's not a clear answer for that, but it may be just healthy behaviors overall. Less smoking, more exercise, maintaining weight, because obesity is a risk for not just diabetes, but for malignancy as well. And there's a long list of malignancies that are associated with obesity. We think about endometrial cancer as one where you never see it in a non-obese person. But colon cancer and breast cancer are both associated with obesity or higher risk. So overall health behaviors are probably a big part of it. It's not just running. There are other things that we don't know very much about that happen when you exercise regularly. You guys probably are well aware of inflammatory cytokines, but there are other related proteins called myokines that are actually secreted from muscle in trained athletes and after bouts of exercise that are beneficial, that actually improve insulin action and have additional anticipated benefits. And we don't know a lot about these. There was an article in the New England Journal last week talking about myokines in response to exercise. Other questions? I guess I've answered everything you could ever want to know then, right? All right, we're good.